It's time we all celebrate the mouse. Let's knock out and knock down Parkinson's disease. Today, mice are more important than ever to research. 40 Nobel Prizes can be directly tied to mice. Mice are very popular model organisms. They are commonly used in research for all these reasons. During the 1980s, mouse genetics underwent a revolution. These experiments culminated in the creation of the first knockout mice. The first recorded knockout mouse was created by Dr. Mario Capecchi. He was awarded the 2007 Nobel Prize in Medicine. Dr. Capecchi's 1987 landmark paper in Cell and his 1989 paper in Trends in Genetics both describe gene targeting to generate mice of a desired genotype. So, what is a knockout mouse? It is deleting a gene or disrupting a gene's function in a mouse. Let's say we have a gene of an unknown function. We will call our gene ABC. To figure out what ABC does, we can look at genes with similar sequences, but we can't do this if nothing is similar. One way to investigate the function of a new gene is to remove that gene and then observe what happens. So, how do we remove the gene? Imagine putting on a pair of boxing gloves and punching out a hole in the mouse's genome. We are effectively knocking out that gene's function. So, how do you create a knockout mouse? Step 1 is to create a targeting vector. The target gene is the one we want to knock out, but to the left and to the right of the target gene is DNA. Instead of putting ABC in the middle of the targeting vector, we put a selection marker. The selection marker will then be expressed instead of the original ABC gene. Once we have our targeting vector, step 2 is to insert the targeting vector into the mouse embryonic stem cells. These stem cells are grown in a petri dish. When these cells grow, divide, and differentiate, all their daughter cells will also be missing our ABC gene. Once we insert the targeting vector into the embryonic stem cells, it will undergo homologous recombination with our target ABC gene. The pieces of DNA will line up and recombine, giving us our selection marker in place of our ABC gene. This will happen in some cells, but not all. Therefore, step 3 is to find embryonic stem cells which have the ABC gene knocked out. In step 4, we will inject correct embryonic stem cells into a mouse embryo. These stem cells that we transplanted are heterozygous. They will have both the knockout and non-knockout stem cells. Then we will let this embryo grow up in the mommy mouse. In order to get the mouse that is homozygous, we will take the babies that are born and use them in the next step. Here in step 5, we will mate mice to obtain a homozygous mouse. In step 6, we will look for all types of phenotypic changes in the knockout mouse. What we will observe will tell us what the ABC gene actually does. I am now going to discuss knockdown mice. In knockout mice, the mice are bred to have at least one particular gene completely erased using DNA. In knockdown, gene expression of a certain specific gene is reduced, but the gene is still very much intact. RNA is inactivated for a specific gene. In June of 2020, very exciting research was published utilizing mice for Parkinson's disease. More than 10 million people worldwide have the brain disorder Parkinson's. My great-aunt has Parkinson's. She was diagnosed in 2014. You might also recognize some of these celebrities who have been diagnosed with Parkinson's. Most people with Parkinson's first develop the disease around age 60. Parkinson's symptoms begin gradually and get worse over time. Listed here are some of the hallmark symptoms of Parkinson's. Parkinson's disease occurs when neurons in an area of the brain that controls movement become impaired and/or die and produce less dopamine, which causes the movement problems of Parkinson's. There are no medical tests to detect Parkinson's. Diagnosis is based on the following. There is currently no cure for Parkinson's. Some therapies can relieve some symptoms. The main therapy is levodopa. Nerve cells use levodopa to make dopamine. Back in 2013, Dr. Zhang Dongfu and his team at UC San Diego were studying the PTB gene. They knocked down PTB using short interfering RNA and fibroblasts. They grew and checked on the fibroblasts in petri dishes. It contained mostly neurons. More recently, Dr. Fu wanted to see if this method could be used to treat Parkinson's in mice, this time using astrocytes instead of fibroblasts. They found that 30% of the astrocytes turned into neurons. These newly generated neurons seemed to grow normally and movement function was completely restored. The mice did not show any symptoms of Parkinson's for the rest of their lives. This research is published in the June issue of Nature. This treatment opens the door to a new therapy which may one day reverse Parkinson's in people. All this is thanks to the amazing mouse. It's time we all celebrate the mouse. Thank you for listening.